Well, my dear students, today's video class is pertaining to the most important part, that is cell division. We know already in various classes you have studied cell division. What is cell division? And you know the the definition of a cell. Cell is a structural and functional unit of an organism. When all organisms start their life journey, so all organisms start their life journey from a single cell, that is zygote. And zygote, single celled structure, undergoes repeated cell division and forms an organism. So cell division is nothing but the growth. So the growth of the organisms takes place by means of cell division. So such a type of cell division we are studying now. And now, types of cell division. How many types of cell division are there? So here, cell division. So there are two types basically. That is, first one is called as somatic cell division, and another one is called as gametic cell division. Somatic cell division and gametic cell division. Two types of cell divisions are existing in nature. So what is somatic? Somatic means other than reproductive. So other than reproductive cell division, for example, body cells. If the growth takes place by means of cell division, that is called as somatic cell division, and that is usually termed as mitosis. Mitosis is nothing but the somatic cell division. It is also called as equational cell division, because when the cell division takes place, the daughter cells which are produced, so they are structurally and functionally equal. They are same. So that type of cell division is called as somatic cell division, and another name for somatic cell division is mitosis. And gametic cell division, which is taking place only in the reproductive organism. For example, in case of plants, in the anthers or in the ovules. In case of animals, in the reproductive structures like testis and ovary, so there only it is confined to the reproductive areas, and that particular cell division is called as the gametic cell division, and it is called as meiosis. Two types of cell divisions are existing here, and these two types of cell divisions lead to the growth and reproduction in organisms. And the organisms which will not reproduce by sexually, they have only one type of cell division, that is mitosis or somatic cell division. This is absent. And the organisms which are reproducing by the sexual methods, so those are having both the type of cell division. One is confined to the body cells, another one is confined to reproductive cells, or for formation of male and female gametes. Such thing is called as the cell division. And now, cell division as a whole, if you take cell division. So cell division is again divided. So of course, somatic and gametic is over. But in somatic and gametic cell division, what are the different types? First one is called as karyokinesis. And another one is called as cytokinesis. Karyokinesis and cytokinesis. The entire cell division has got two steps. First step is karyokinesis. Second step is called as cytokinesis. So what is meant by karyokinesis? Carry on. Two words are there here. Carry on stands for the nucleus. So the division of nucleus is called as karyokinesis. And when for example, this is the mother cell. It has got a nucleus here, and during karyokinesis, the nucleus undergoes division and forms two nuclei. So this is called as karyokinesis. Karyokinesis is the division of nucleus, and after the division of nucleus, the cytoplasm present in the mother cell is divided. So there is formation of a wall here, separating wall. So this wall is formed, that is a division of cytoplasm. That is called as cytokinesis. Karyokinesis and cytokinesis. Such two daughter cells are produced here. Such daughter cells are structurally, functionally are equal, same. That is why it is called as equational cell division. So karyokinesis and cytokinesis. Now, karyokinesis, in case of mitosis, I am telling you now. So today we are discussing about mitosis. In mitosis, karyokinesis is divided into 5 stages. First one is called as interphase, and prophase, metaphase, anaphase, and the last one is telophase. That is called as telophase. So interphase, prophase, metaphase, anaphase and telophase. So these are the different stages of cell division only which are occurring in karyokinesis. These 5 stages are nothing to do with cytokinesis. Let us discuss later. So this is called as the karyokinesis. So now let us go one by one. What is the first cell division? That is called as interphase. Okay. What is interphase? In between. In between 2 cell division, this is the phase. So after the cell division, after the division of a mother cell, 2 daughter cells are produced. These 2 daughter cells will enter into next stage of cell division. So that is in between these 2 cell division, there is a stage or phase what is known as interphase. So earlier days, so it was presumed that interphase is the resting stage. Cell will not do anything. So it undergoes rest, and after resting it enters into second stage. But after the continuous research, it was found that it is not the resting stage, it is the highly active stage. Very active stage it is, because in this interphase there is preparation for the next division. The cell prepares for the next division. So without interphase, the cell cannot enter into interphase, the next cell division. So this is called as interphase. The interphase is having G1, then S and G2 phase. So these 3 types of stages are there: G1 phase, G2 phase, sorry, S phase and G2 phase, and after which you will find the cell division phase. So these are the 4 different stages occurring. So cell division we shall not consider here. So interphase is containing G1, S and G2 phase. So this is in these 3 stages the cell is preparing for the next cell division. So that is called as the interphase. And interphase is having about 12 hours duration. G1 phase has got 12 hours during which protein biosynthesis, RNA synthesis takes place. So whatever the RNA, DNA is required for the next division, that is synthesized in the G1 phase. And S1 phase, the DNA synthesis takes place. It will be of 8 hours duration. I am telling about interphase. And G2 phase, it is again 12 hour duration, during which again RNA, DNA process is taking place. So huge amount of RNA, proteins are synthesized and kept in the cell for the use of second mitotic division. So this is called as interphase. Totally the interphase is taking about 28 hours. Please remember, 28 hours is the interphase duration. So when one cell is completed, telophase is over. So next it enters into interphase, and interphase completes, it requires 28 hours for the completion. So for 28 hours it prepares and it enters into next cell division. And the next cell division is what? Prophase, metaphase, anaphase, telophase. These 4 stages are there. All these 4 stages require only 1 hour. So we have to learn here, for 1 hour process, the cell is preparing for 28 hours. So this is the fun of cell division. So this is called as interphase. Let us see the details in the cell cycle. Interphase. So okay, in the interphase, the cell is preparing for the cell division. It synthesizes huge amount of protein, huge amount of RNA and huge amount of DNA in the form of replication. So everything is over. So now the cell is ready to enter into cell division. First stage is called as First stage is called as prophase. Prophase. In the name, pro stands for before. So before entering into actual cell division, what is the stage? That is called as prophase. So it is the first stage of cell division, where I will just draw the diagram here. So this is the cell immediately after interphase. So it has got the nucleus and it has got cytoplasm and so many cell organelles, and inside you are finding the chromatin network, and you will also observe the nucleolus. So this is the nuclear membrane, and this is the nucleolus, and this is chromatin network. You will not find the chromosomes, and you will find the nuclear membrane and cell division, everything is cell organelles, everything is here. So when the cell wants to enter into next cell division phase, so prophase begins. This is called as early prophase. And late prophase, what happens is, first thing, the disappearance of the nuclear envelope. Nuclear envelope disappears. Gradually, it is withdrawn by the Golgi apparatus, and these Golgi apparatus, they withdraw their membranes, so that the nucleolus gradually disappears, and ultimately it totally disappears. First thing. Second thing is, the cell absorbs water and the protoplasm becomes rich. And third one is, nucleolus gradually disappears. And the chromatin network, which is very long DNA fiber, that undergoes condensation. And because of the condensation, so for example, I will just draw the late prophase. So here you will find the chromosomes are produced here. So this is the structure, which are very, very thick, and these are the things. No envelope here, no nucleolus. So all the chromosomes are shed in the cytoplasm. So by this, the prophase is over. I will repeat once again. Prophase is the stage where the cell absorbs water from the surrounding, and it becomes rich in protoplasm. And when it becomes rich in protoplasm, first thing is the chromatin network undergoes condensation, and thick, stout, rod-like chromosomes are developed. Second one, nucleolus gradually disappears. At the end of prophase, the nucleolus totally disappears. And nuclear envelope starts disappearing, and at the end of prophase, and it also disappears. So this is the one. And at the end of prophase, from the polar region, you will find the production of innumerable thread-like structures, and these thread-like structures are called as spindle fibers. Spindle fibers. Spindle fibers are produced. And these spindle fibers, they grow, and they come toward the center. Say, for example, this is the chromosome. So this is the chromosome here. Okay. And spindle fibers are produced here. And these spindle fibers, they come and hook the centromere. So, and from this pole also, the spindle fiber will come and hook the centromere. So this is the spindle fiber. Spindle fibers are produced, and these spindle fibers, they come and hook or attach to the chromosome. At what place? Only at the centromere position. And if there are about 8 chromosomes, so spindle fibers will not be produced only 8. They are produced in so many number. But some of these spindle fibers will not get the chromosome, because each centromere will receive only one spindle fiber from opposite poles. So because of this, some spindle fibers will not get the chromosome, and they become attached here, and these are called as polar fibers. These are called as polar fibers. So this is the end of prophase. Okay. Now, when the spindle fibers, they are developing here, so I have told you already, they are produced, and they come and hook the centromere. And when the centromere are hooked, the spindle fiber, they start pulling the chromosomes. But these spindle fibers, 2 spindle fibers from this pole and this pole, they exert equal pressure on the centromere. So that when the pulling is equal, all the centromeres, they are settled at the middle portion of the this one, that is the cell. So this is called as metaphase. Metaphase. In the metaphase, what happens is, because these spindle fibers are attached to this, so for example, so these are the chromosomes. So this is the chromosome here. So I will just draw the chromosome. So these are the chromosomes, and these chromosomes, they become settled at the equatorial position. So you can find here. So these are the spindle fibers, and these spindle fibers will hook the centromere, and they start pulling the chromosomes, and because of the equal pressure or equal force from both the sides, so the centromeres of each chromosome, all the chromosomes, they are shuttled at equidistant from both the poles. And this position, so this is called as this one, and this position or this one is called as equatorial plate. Equatorial plate. Where, see, what is present at the equatorial plate? So many a times what you tell is chromosomes are present at the chromosomes are not present at the equatorial plate. Only the centromeres are present at the equatorial plate. Okay. So they are present at the equatorial plate. So this is called as metaphase. Meta means middle. So middle phase of the cell division, where the chromosomes are very much thick, they are stout, and the centromeres are hooked by the spindle fibers from opposite poles, and when they are pulled apart from both the poles in equal pressure, so all the centromeres, they start settling into the midpoint of the cell, or you can say equidistant from both the poles. And this stage is called as metaphase. During this stage, the chromosomes are very thick and condensed. For the study of morphology of chromosomes, always the metaphase chromosomes are preferred, because in the metaphase, the chromosomes are very much thick, they are very much condensed, and the morphology is very clear. So this is called as metaphase. Now coming to anaphase. Anaphase. Anaphase means towards the end of the stage. Towards the end of the phase. What happens here? I will just show you. This is the centromere, and this is the chromosome. Okay. And from this one, two spindle fibers are attached here. So these are the spindle fibers. This is the chromosome. What happens is, these spindle fibers will not sit quiet. They start continuously pulling the chromosome. So from opposite poles, equal pressure or equal force is applied on the centromere. Centromere will sustain the pressure up to some extent. A stage will come. At that time, the centromere becomes broken. So centromere breaks. Say for example, this is the centromere, two centromere, and one arm goes to this side and another arm goes to this side. So centromere is broken now. When the centromere is broken, these are called as chromatids. This is chromosome. After the breaking of the chromosome, two chromatids are produced. And since they are attached by this spindle fiber, this chromatid will start moving towards this pole, and this will start move towards the opposite poles. So if there are about 8 chromosomes, 8 chromatids are moving towards this pole, and 8 chromatids are moving towards this pole. So I will just draw the diagram here. 1, 2, 3, 4. So this is the chromatid here. So this is the chromatid. So, and here. So these are the chromatids. Okay. So these are the chromatids, and this is the pole here. So all the chromatids, they start moving towards opposite poles, and this is called as anaphase. What is anaphase? So when the anaphase begins when the centromere breaks. As soon as the centromere breaks, anaphase starts. So depending upon that, early anaphase, middle anaphase, late anaphase, depending upon the author, we can see the things here. But anyway, when the centromere of the chromosome breaks due to the pressure exerted by the spindle fiber, the centromere breaks and the two chromatids are produced, and each chromatid, they are moving towards opposite poles because they are still attached by the spindle fibers. So spindle fibers pull the chromatids towards the opposite poles. So the chromatids start moving towards opposite poles. This stage is called as the anaphase. Anaphase is the movement of chromatids towards opposite poles. Anyway, depending upon the shape of the chromosomes, if it is metacentric, you will find V-shaped structure. If it is submetacentric, it is L-shaped. If it is acrocentric, it is called as J-shaped, and if it is telocentric, it is formed I-shaped, depending upon that. So anyway, so these spindle fibers are produced here, and these spindle fibers, they pull the chromatids towards the opposite poles. Once these chromatids reach the opposite poles, for example, so I will just show you here. So the chromatids will move towards this stage now. The chromatids have come and they have reached the poles now. See, for example, so 4 chromatids here. These are the chromatids actually. Chromatids are produced. So these chromatids have reached the poles now. So when the chromatids have reached the poles, anaphase is over. Another stage begins. What is that stage called? Telophase. What is telophase? Telo means last. Telo means end. So end phase is called a telophase. Prophase means what? Beginning stage, first phase. And meta, middle phase. Anaphase, towards the pole the chromatids are moving, that is called as anaphase. Telophase means when the chromatids reach the opposite poles, so telophase begins, and this telophase is the last stage of mitosis. Anyway, mitosis is over now. When the chromatids reach the opposite poles, whatever has happened in the prophase, opposite things happen here. What was happened in the prophase now? First thing is the chromatin network undergoes condensation, and the chromatin network becomes into thick, stout chromosomes. But here already the thick and stout chromatids are there. Now decondensation begins. Thick chromosomes are decondensed and they are producing the what you call the chromatin network. And the nuclear envelope starts appearing gradually. Nuclear envelope starts appearing gradually. It appears. Okay, and I will draw the another diagram here. Another telophase after this. So the nuclear envelope becomes very much pronounced. So you can find here nuclear envelope. And inside the chromatid, thick stout chromatids are there. So these chromatids are becoming decondensed, and because of the decondensed condition, it becomes again the chromatin network. What was present during the prophase, the same thing begins here. So this is called as the chromatin network is produced. Okay, and in the prophase, the nucleolus disappears. So here the nucleolus reappears. Nucleolus reappears. Okay, by this 2 daughter nuclei are produced now. So from one nucleus, 2 daughter nuclei are produced, and this phenomenon is called as karyokinesis. Karyokinesis is over. And after this karyokinesis, another begins now, another division takes place, that is called as cytokinesis. What is cytokinesis? Division of cytoplasm is called as cytokinesis. So how it starts? In case of plant cells, I am telling, I am not concerned about animal cells. I will just tell you what happens. After the completion of this telophase, at the equatorial plate region, what is the equatorial plate region? During metaphase, where the chromosomes, the centromeres of the chromosomes were settled. So that area, for example, this area is called as the cell plate region or equatorial plate region, equatorial region. In this equatorial region, a thick stainable portion is produced. And this is called as cell plate. That is called as cell plate is produced. Cytokinesis I am explaining with respect to plant cell. Prophase, metaphase, anaphase, telophase, they are same in both animal cell and plant cell. But cytokinesis varies from plant to animal cell. I am explaining now cytokinesis in plant cell. So after the completion of telophase, a cell plate is appearing, and this cell plate starts growing on this side and this side. Ultimately, the cell plate reaches the opposite walls. Understand now? So cell plate grows on opposite sides. It is just a cell plate now. It is not a cell wall. And on this one, the secondary wall material, the wall materials are deposited on both the sides. That is cellulose is deposited. And this cellulosic material deposited, and now there is formation of a cell wall. Cell wall is produced now. So this is the cell wall, and the first layer, what was the bifurcation layer, is produced in the cell that forms the middle lamella. And on the either side of the middle lamella, cell wall is synthesized. How is it synthesized? The cell starts depositing cellulose on both the sides of the middle lamella, and a cell wall is produced. By this, two cells are produced. Two daughter cells are produced. And these two daughter cells have shared equal number of cell organelles, equal number of chromosomes in quality and quantity wise. And because of the equal sharing of all the cell contents of the cells of the mother cell, so the mitosis is always called as the equational cell division. So that is called as homogeneous cell division. So this is the end of prophase, metaphase, anaphase, telophase. Okay. Now I will discuss one more important aspect here, what is known as the cell cycle. Very important. Cell cycle. Okay. When I was discussing about interphase, I had discussed G1, S, G2 and cell division. Now I want to tell you now, what is the duration of G1? What happens during G1? What is the duration of S? What happens in the S during S? And what is the G2? What is the duration and what happens during that? Because this G1, S and G2 are not the silent phases. They are not the resting stages. They are very active stages, active phases. And what is this one? Let us see cell cycle now. If you draw the cell cycle here, this is called as cell cycle. And here I will just show you like this. Okay, this is called as this one. And this is called as G1 phase. This is called as G1. This is called as S. And this is called as G2. What is G1? So G1 needs 12 hours. 12 hours. Yes, 8 hours. And G2 also needs 12 hours. Please remember. And this is called as mitosis. Mitosis or meiosis. So only this much. How much it requires? It requires 1 hour only. 1 hour. Oh, for 1 hour work, the cell is preparing for 28 hours. So the G1, S and G2 all together called as interphase. Interphase, see, interphase, prophase, metaphase, anaphase, telophase. 5 stages we are telling. Out of this, which is the longest stage? Longest stage is called as interphase. Interphase is nothing but, interphase is the preparatory stage for the cell division. It has got 3 substages, G1, S and G2. So what the cell does during G1? G1 stage, the cell is preparing RNA and protein. RNA and protein are, they are produced in plenty. Because whatever RNA was there in the mother cell, that has been distributed to daughter cells. Huge sufficient quantity of RNA proteins are not there. So the cell is now preparing for RNA and protein. And here, as compared to mother cell, 50% of the DNA is transferred to this daughter cell, 50% to this daughter cell. So with this 50%, the cell is not complete. So the cell wants to become complete means, and here DNA synthesis takes place. That is called as replication takes place during the second stage. Replication takes place during the second stage, and here also again RNA and protein. Okay. Now a question comes here. So after the completion of mitosis, G1 stage begins. After telophase, what is the first stage? G1. In the G1, we are told that there is already production of RNA and protein, and after RNA and protein, the S stage, that the DNA, the replication takes place. Again, why do you need G2 phase? Already the RNA and protein are synthesized. Why you need RNA and protein synthesis in G2? So the answer is very simple. RNA and proteins are synthesized sufficiently, no doubt, but these RNA and proteins are somewhat used in the second stage. During the synthesis of DNA, what is known as replication, what amount of RNA is there in the cell, that particular some amount is utilized already. So that remaining RNA and protein are not sufficient for the cell division purpose. So again G2 phase comes, and in this G2 phase again RNA and protein are synthesized. And after this one, 12 plus 8 plus 12, 28 hours preparation is already over, and now the cell is ready for mitosis, prophase, metaphase, anaphase, telophase. All these 4 stages are taking place. So the duration of mitosis is only 1 hour. Within 1 hour, so the entire thing is completed. Totally, one cell cycle. Cell cycle means prophase, metaphase, anaphase, telophase, interphase. All these 5 stages totally need how much time? They require 29 hours. Once cell division is over, 28 hours preparation, 1 hour cell division, again 28 hours preparation, 1 hour cell division. Like this, the things are continued. So this is called as the cell division or mitosis. And what is the significance of mitosis? So significance of mitosis is only one, that is the growth. When we are growing, it is because of mitosis. When the mitosis is taking place in our body, that shows that we are growing. So that is called as, in case of plants, mitosis takes place in meristematic cells. Meristematic cells we have studied in anatomy already, that there is apical meristem, lateral meristem, intercalary meristem. All these things we have discussed already. So the cell which are actively dividing, the tissue which is actively dividing is called as meristematic tissue. Meristematic tissues are showing the active stage of cell division. So this is the class for today, and tomorrow we shall discuss about another type of cell division which is very important, that is gametic cell division, and it is also called as the meiosis. Meiosis is little bit complicated. So please, you go through the notes, you go through the video class of today and be prepared for the next class. So before becoming perfect in the mitosis, don't go to meiosis, because the concept of cell division is same. In meiosis also same prophase, metaphase, anaphase, telophase, same thing are there. What is happening in prophase here, same thing happening there also. What is happening in mitosis of this metaphase of mitosis, same thing happens there. But the order of the sequential order changes. The different things are coming in between, and those things we shall discuss tomorrow. And be attentive and study well. And whatever problems we have, of course, none of you are asking me the questions. But anyway, if you have doubt, any doubt for that matter, you ask me anytime, you contact me anytime. You have my cell number, contact me, and you can discuss. And shortly we may receive the timetable from university. So as soon as I receive, of course, all staff members are attentive and they will put in your group. So accordingly, you have to prepare. So thank you. Have a nice time.